Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, so we've now discussed many examples of calcium signaling which involve calcium being released from the intracellular stores, so the endoplasmic reticulum or sarcoplasmic reticulum in muscle cells, okay, and then going into the cytoplasm and producing these calcium signals. Okay, what I now want to do is turn my attention onto an example of where it isn't being generated from intracellular stores, where instead the calcium that comprises the calcium signal is coming from extracellular fluid calcium. Okay, so channels in the plasma membrane this time are going to open and allow calcium to come into the cytoplasm of the cell, creating a calcium signal. Okay, and that will have the effects within the cell, basically. So remember, there is that very powerful gradient of calcium across the cell membrane. Okay, we have a calcium concentration of 1.5 millimolar extracellularly and 100 millimolar intracellularly, a 15,000-fold gradient favoring the movement of calcium in. So when you open channels for calcium in the plasma membrane, you do get calcium moving in, basically, and that will create a calcium signal within the cytoplasm. Okay, right. So, the prototypical example of calcium signals that are generated from extracellular fluid calcium, which is really physiologically important, is synaptic vesicle exocytosis. Okay, so basically, in the axon terminals of neurons, neurotransmitter is released from the axon terminals when action potentials arrive in the axon terminal. So when the neuron fires an action potential, it will propagate along the axon into the axon terminal, and that's going to trigger the axon terminal to release neurotransmitter. Okay, the question is how? What is actually occurring? And this involves a calcium signal being generated in the cytoplasm of the axon terminal, which drives the release of structures called synaptic vesicles, which store neurotransmitter, basically. So this is a process called synaptic vesicle exocytosis. So exocytosis is the fancy way of saying release of the contents of synaptic vesicles into the extracellular fluid. Okay, right. So firstly, let's talk about synaptic vesicles and what they are. So where is the neurotransmitter being stored before it's released? Because usually in axon terminals of neurons, the neurotransmitter has been pre-made before the action potential arrives, okay? And then all you're going to do is release it into the extracellular fluid once the action potential arrives. So let's talk about the storage of neurotransmitter in synaptic vesicles. So here, this is our axon terminal here, okay, of some neuron. Okay, so this is the axon here, and then this bulge is the axon terminal. Okay, now in the axon terminal then you have machinery for producing whichever neurotransmitter this axon terminal releases, be it glutamate, acetylcholine, GABA, whatever. Okay, we'll just generalize this and call them all neurotransmitters. Okay, and basically what happens is the neurotransmitter is packaged into membrane-bound structures called synaptic vesicles. Okay, so this is going to represent our synaptic vesicle here, and what we're going to do now is package our neurotransmitter into that synaptic vesicle. So I'll represent the neurotransmitter by these green dots. Okay, so the neurotransmitter gets packaged into the synaptic vesicles. Okay, so neurotransmitter NT is going to go into the synaptic vesicles. Then, once the synaptic vesicle is full of neurotransmitter, what happens is they are attached to and primed at the membrane where they're going to be released. Okay, so the membrane which actually faces the membrane of the postsynaptic cell is called the presynaptic membrane. Okay, so what I've now highlighted up in orange here, this is representing the presynaptic membrane here. Okay, and this will be facing the postsynaptic membrane of the postsynaptic cell here, which will have the receptors for our neurotransmitter on. Okay, so what happens in the axon terminal is that these synaptic vesicles that you've created are going to be attached and primed, and I'll explain exactly what primed means in a moment, to the presynaptic membrane here. Okay, so they are attached and then primed. So attaching, hopefully you understand what attaching means. Okay, but what does priming mean? Okay, priming means that these synaptic vesicles that are attached to this presynaptic membrane and primed there are now ready to be released as soon as an action potential arrives in the axon terminal. So when an action potential arrives in the axon terminal, and I'll just abbreviate action potential to AP, it's going to trigger some activity in the axon terminal, and I'll let the cat out of the bag now. It's going to be a calcium signal coming into the cytoplasm of the axon terminal that will then trigger the release of that primed synaptic vesicle. So you're not just attaching the synaptic vesicle to the presynaptic membrane, you're priming it, which means that it's ready to respond to the arrival of an action potential, and it's ready to fuse the instant the action potential arrives. Okay, so you could just attach it there, but that doesn't mean that it's ready to be fused once an action potential arrives. So priming refers to the fact that it is now ready to actually fuse the instant an action potential arrives in the axon terminal. And then, of course, what will happen is it will release its neurotransmitter into the synaptic cleft, the space between the presynaptic membrane and postsynaptic membrane, once it has fused with the plasma membrane. So the membrane of the vesicle becomes part of the plasma membrane, the lumen of the vesicle becomes part of the extracellular fluid, and the neurotransmitter will be released into the synaptic cleft. Okay, right. So, what we want to study then is how does the action potential cause the exocytosis of these primed synaptic vesicles, basically. Now, to understand that, we firstly need to understand how these synaptic vesicles are primed onto the presynaptic membrane. So, what actually comprises this priming? Okay, now, I should just say that this field is still evolving, okay, it's not set in stone, but what I'm going to tell you is the very much so believed theory of how this works, okay, but it isn't set in stone, it may well evolve, okay. So the basic idea here is that these primed synaptic vesicles, you are going to have proteins that are in the vesicle membrane attached to proteins that are in the presynaptic membrane. Okay, and these proteins are going to be holding the two membranes, the vesicular membrane and the presynaptic membrane, very close together, basically. Okay, and then somehow when the action potential arrives, those proteins are going to pull the membranes even closer together, and then the two membranes are going to fuse. That's the basic idea. Okay. So, let me now tell you about these proteins that are in the vesicular membrane and are in the presynaptic membrane, which are involved in attaching the vesicle to the presynaptic membrane, basically. Okay, so these proteins that are involved in doing this, they are called snare proteins. Okay, so they're all going to be snare proteins, and there's 3 snare proteins that we're going to see that are going to be involved in this. Okay, now what's the characteristic feature of a snare protein? What do you have to be considered a snare protein? Well, snare proteins all have a very important motif, basically, a little portion of them that they all have in common, which you have to have in order to be considered a snare protein. Okay, and this is called the snare motif. Okay, now the snare motif is a great long alpha helix, basically. Okay, and we'll see what the function of this alpha helix is later, but I'll just draw a little picture of this here. Okay, so all of the snare proteins are going to have these snare motifs, which are alpha helices. Okay, and what in fact is going to actually happen, I'll let this out now, okay, is that the snare proteins in the vesicle membrane and the snare proteins in the plasma membrane, their snare motifs are going to intertwine with one another. The alpha helices are going to coil around one another in a coiled coil-like way, okay? And they're going to form a great bundle, which is what's going to hold the vesicular membrane to the plasma membrane. Okay, and I'll explain that more later on, but for now know that that's what connects all of these snare proteins together. They all have these snare motifs, these alpha helices. Okay, the other thing that you should know before we actually go into the details is that there are 4 different types of snare motifs. Okay, and these 4 different snare motifs are called the QA snare motif, the QB snare motif, the QC snare motif, and the R snare motif. Okay, and in fact, when you form this bundle of snare motifs, it's going to involve 4 snare motifs, and it's going to involve one of each of the 4 types, basically. Okay, now I've said that we're only going to have 3 snare proteins, but then 4 snare motifs, that doesn't seem to add up. Well, one of the snare proteins is going to actually contain 2 snare motifs, as we'll see. Okay, so when you form these complexes between proteins in the vesicular membrane and plasma membrane, overall they're going to involve 4 snare motifs in 3 snare proteins, okay, and they're going to form a 4 helix bundle, which is what's holding the 2 membranes together, basically. Okay, right. So let's now actually talk then about the snare motifs, about the snare proteins that are involved in this process. So you can divide the snare proteins that are involved in this process up into 2 different classes. Okay, and the division is very simple. There are the T snares, okay, and the T snares are the ones in the plasma membrane, okay, and the T here stands for target. So the snare proteins that are in the plasma membrane and are involved in this joining between the plasma membrane and the vesicular membrane are called the target snares, or the T snares. Then there are the snares which are in the vesicular membrane, and these ones are called the V snares, okay, for vesicular snares. Okay, so the V here is for vesicular. Okay, and we're going to see that in the vesicle membrane you only have 1 type of snare protein. Of course, you have multiple copies of that snare protein in the vesicle membrane, but there's only 1 type of V snare in the synaptic vesicle membranes. Whereas in the plasma membrane there are 2 different types of snare protein inhabiting the plasma membrane. Okay, right, so let's start by talking about the T snares, and then we'll come on to the 1 V snare. Okay, right, so the first T snare then that I want to talk about is a T snare called syntaxin 1. Okay, so this is a snare protein. It's going to contain a snare motif, and it's a T snare, so it's going to be within the plasma membrane. So let me now draw you a picture of syntaxin 1. Okay, so I'll start by drawing the plasma membrane then. So this is representing the presynaptic portion of the axon terminal membrane, basically. Okay, now I'm going to have this oriented in an odd way. Okay, this is going to be the extracellular fluid below this time, and this is going to be the cytoplasm above this time. Okay, so whenever I've been drawing plasma membranes before, I've always had it oriented the opposite way. So be aware that I've suddenly decided to switch around and have the cytoplasm on top and the extracellular fluid on bottom. Okay, right, so let's now draw syntaxin 1. So it's an integral membrane protein. It's going to have a membrane-spanning alpha helix, which is holding it in the plasma membrane. And the important portions of it are going to be on the cytoplasmic side. So here's its amino terminus. Okay, then you have an important domain consisting of 3 alpha helices bundled together. Okay, then you have the snare motif, which is going to be this portion here. Okay, then you have the membrane-spanning alpha helix, and then the C-terminus is over here. So let me colour in these different domains. So here in orange, these are representing 3 alpha helices, 3 little alpha helices that bundle together. Okay, and this little domain of 3 alpha helices bundled together over here, this is known as the HABC domain, okay, because H for helix, and then one of the helices is called the A helix, the second is called the B helix, and the third is called the C helix. So it's the HABC domain made up of the bundle of those 3 different helices. Okay, then after the HABC domain, you then have the much bigger alpha helix, which is really important, okay, which is a snare motif alpha helix. And this specifically is a QA type snare motif represented in red here. Okay, then of course you'll have another alpha helix, which will be the membrane spanning alpha helix there, but I won't colour that in. Okay, so this is syntaxin 1 then. We've seen its snare motif. It's got this QA snare motif, and that's the portion that we're going to be really interested in. That's the portion that's going to play an incredibly important role. Okay, right. So that's syntaxin 1. That's one of these T snares here. Next, let's talk about the other T snare present within the presynaptic membrane. Okay, and the second T snare that I want to talk about is called SNAP25. Okay, right, now SNAP25 is the snare protein that has two snare motifs. So I said that when we form one of these snare complexes involving the T snares and the V snares, it's going to have 4 snare motifs all coming together to form a 4-helix bundle. Okay, so 3 snare proteins, 4 snare motifs. One of them must have 2 snare motifs, and that's the one. This is SNAP25 that has these 2 snare motifs. Okay, so SNAP25 isn't a membrane-spanning protein. Okay, it's not an integral membrane protein. However, it is attached to the plasma membrane by lipid moieties that have been attached onto it, basically. Okay, now, it is within the cytoplasm then, so I'll show it here, like so. So this is how I'm going to draw SNAP25. Here's its amino terminus, here's its C terminus, and then here it's going to have multiple lipid moieties attached onto it, which are palmitoyl groups for those who are interested, okay, which are going to implant into the, whoops, oh no, that's fine, sorry. Once again, let me just make sure that you understand this is the cytoplasmic side and this is the extracellular fluid side. I was just thinking the ECF there, no, that's obviously referring to this picture. So imagine moving those labels down to here. This is the cytoplasmic side and this is the extracellular fluid side. Don't let that confuse you. Okay, so those lipid moieties then attached to the SNAP25 protein are going to implant into the inner leaflet of the phospholipid bilayer and hence hold SNAP25 attached to the inner leaflet of the phospholipid bilayer. Okay, so these 2 regions then are the 2 snare motifs of SNAP25, and they are different types of snare motifs. So this first one here is called the QB snare motif. This second one here is called the QC snare motif, okay, of SNAP25. Right, so there are those 2 snare motifs in SNAP25. So now what I want to show you is how syntaxin 1 and SNAP25, even before the vesicular snares have come along, can form a complex called the T-snare complex. So let me show you this T-snare complex. Okay, so these form in the presynaptic membrane before the synaptic vesicle has come along, and then what will happen is the synaptic vesicle will contribute its T-snare, which we'll study in a moment. The synaptic vesicle will contribute its V-snare, which we'll see in a moment, okay, and that will form the full snare complex. Okay, so let me now show you the T-snare complex then. So here is the presynaptic membrane again. This is the cytoplasmic side, the extracellular fluid side. I'll once again draw syntaxin 1 like so. Here is the C terminus here. Okay, I'll colour in the alpha helices of the HABC domain in orange there. Here is the QA snare motif. And then what we'll have is the SNAP25 protein. Its snare motifs will now be intertwined with the snare motif of syntaxin 1 here, so they'll be coiled around one another basically. Okay, now I haven't attempted to show them actually coiled around one another because it makes the picture look messy, but imagine these all twisted around one another into a bundle basically, and that's what you've got there. Okay, and there are those lipid moieties attaching the SNAP25 to the inner leaflet of the plasma membrane. Okay, right, so here we have this complex between the 2 T-snare proteins in the presynaptic membrane, syntaxin 1 and SNAP25. Okay, now what's going to happen is we're going to bring in the V snare. So I'll go over the page and we can talk about the V snare. So the snare protein now that's in the synaptic vesicle membrane. Okay, so there's only one type of V snare actually in the synaptic vesicle membrane. Okay, and this is called synaptobrevin 2. Okay, now synaptobrevin 2 also has another name that you might come across. It's also called VAMP2. Okay, and that stands for Vesicle Associated Membrane Protein 2. Okay, so the V is for vesicle, the A is then for associated. Okay, the MP is for membrane protein, and then obviously the 2 is just for 2. So vesicle-associated membrane protein 2. So if you do see people referring to VAMP2, don't be afraid, that's just synaptobrevin 2. Okay, which is this really important V snare. Okay, so let me now draw synaptobrevin 2 within the vesicle membrane. Okay, so I'll firstly draw a synaptic vesicle here. So here is the synaptic vesicle membrane, and once again it's a phospholipid bilayer, so I'll draw it as a phospholipid bilayer here. Okay, so obviously this is the luminal side of the synaptic vesicle membrane, and this is the cytoplasmic side of the synaptic vesicle membrane. Okay, now the amino terminus of the synaptobrevin 2 is on the cytoplasmic side, along with the snare motif, and the C terminus is on the luminal side, like so. Okay, you'll have a membrane spanning alpha helix crossing the synaptic vesicle membrane here. Okay, then you have a snare motif here in purple. Okay, which is also on the cytoplasmic side. And the snare motif of synaptobrevin 2 is an R-type snare motif. So this is my picture then of synaptobrevin 2. It's an integral membrane protein in the membrane of the synaptic vesicle. Okay, its snare motif is on the cytoplasmic side, as are all of the snare motifs of the T snares that we've seen, and it's got this R snare motif. Okay, and its amino terminus is on the cytoplasmic side, and its C terminus is on the luminal side. Okay, so the synaptic vesicle will have lots of synaptobrevin 2 proteins in its membrane. Okay, not just one of them. Okay, now what's going to happen is that when the synaptic vesicle comes to the plasma membrane, you're now going to form loads of snare complexes where the synaptobrevin 2 molecules are going to attach to T-snare complexes that we've seen. Now you're not just going to form one of these snare complexes, okay, you're going to form multiple of them. When you draw a picture of the synaptic vesicle bound and primed to the plasma membrane, often it looks nice if you show 2 of these snare complexes symmetrically positioned relative to each other. However, it could form more than 2, basically. It will form more than 2. Okay, but I'm about now to show you a picture of the synaptic vesicle attached to the plasma membrane by 2 snare complexes. Okay, but be aware that there will be more than 2 being formed. Okay, it's just that this is how the picture looks nicest if you do it like this. So here is the synaptic vesicle here. Here now is the presynaptic membrane. This is the extracellular fluid side, so I might just label this up because this is the big picture now. Okay, this is the cytoplasm here. Okay, and I'll put a little bit of colour onto this picture. So we'll have the cytoplasm and extracellular fluid underlined in green there. Okay, right. So now what I want to show is 2 snare complexes involving A synaptobrevin 2, A SNAP25, and A syntaxin 1 protein each. Okay, right. So we'll start by putting on the synaptobrevin 2 proteins. Okay, so here is synaptobrevin 2. The amino terminus is obviously cytoplasmically located, and the C terminus is on the lumen. Okay, and here are their R snare motifs in the purple here. Okay, then I'll put on the syntaxin 1 here. Okay, here's one syntaxin 1 for that snare complex. Okay, we'll have the other syntaxin 1 over here for this snare complex that we're forming over here. And let's colour in the different portions of syntactin 1. So there is that HABC domain in orange there. Okay, and here is the QA snare motif here in red. And then all I need to now add on is our SNAP25. Oops, meant to show it twisting down here so that I could have the lipid moieties. Never mind, the lipid moieties will just have to extend now. Okay, and here is that SNAP25 there. Okay, here's the N terminus, here's the C terminus, here's the N terminus, here's the C terminus, and then of course you've got the QB and QC snare motifs here, QB and QC. Okay, so basically these 4 snare motifs are all going to coil around one another to form what's called a 4-helix bundle. Okay, and basically it's these 4 helix bundles which drag the plasma membrane and the vesicle membrane very close together. Okay, so let me show you how these 4 helix bundles actually assemble. So basically, it's often compared to a sort of zippering up mechanism, and the zippering up begins at these sort of peripheral ends of the snare motifs, these far ends over here. Okay, so these portions. So you start coiling them round one another from these portions at the ends, and then it spreads basically towards the centre. And as it spreads towards the centre, it brings these 2 membranes closer and closer and closer. And the idea is that if the zippering up was to reach right to the end, these 2 membranes would actually be brought so close together that they would fuse. Okay, but hang on a moment, we don't want them to fuse. We want them to stop just before the end. We want to prime them, we want to get them ready to fuse, but we don't want them to actually fuse until the action potential arrives. So what stops the snare complexes that we've shown here from zippering up all the way to the end and bringing the 2 membranes so close together that they fuse? Well, a final protein is going to come in here, okay, into this core snare complex, or each of these snare complexes here. Okay, and I'll colour this protein in yellow here. Okay, and this protein is called complexin, and it too has an alpha helical portion. Not a snare motif, but an alpha helical portion. Okay, so complexin binds to each of these snare complexes and stops the zippering up proceeding right to the end, so it halts, or the word that people often use to describe this is clamps. It clamps the zippering up of the snare motifs and stops them from zippering up right to the middle, basically, and therefore it allows them to bring the 2 membranes very close together, but not so close together that they will fuse. So if you now want to restore the zippering up process, if you want to finish the zippering up process, what you need to do is remove the complexin proteins here from our snare complexes, and that is what's going to happen when an action potential arrives in the axon terminal, okay? And it's that we now want to study. So what actually triggers these complexin proteins to be removed when an action potential arrives? Okay, so let's bring back up our picture of the entire axon terminal here. So basically, it's calcium. It's all about calcium, and that's why we're discussing this. Okay, calcium signals are going to trigger the removal of the complexin proteins from the 4 helix bundles of these snare complexes. Okay, not the electrical signal. The electrical signal has to be turned into a calcium signal. Now, how can we turn electrical signals into calcium signals? Well, we can use voltage-gated calcium channels. So basically, in the membrane of the axon terminals, you will have voltage-gated calcium channels. And these voltage-gated calcium channels are what are known as PQ-type voltage-gated calcium channels, or they're N-type voltage-gated calcium channels. So we have seen what is meant by L-type voltage-gated calcium channels. Okay, we do not yet know what is meant by PQ-type and N-type voltage-gated calcium channels. However, the meaning is very similar. Okay, so bringing back up our discussion of voltage-gated calcium channels, we talked about how L-type voltage-gated calcium channels all had alpha-1 subunits that were in this CAV1 family of alpha-1 subunits of voltage-gated calcium channels. Now, we didn't then discuss these two other families. So basically, if you make a voltage-gated calcium channel where the alpha-1 subunit is CAV2.1, okay, this one in this second family, then your voltage-gated calcium channel is what's known as a PQ type voltage-gated calcium channel. Okay, so that is what is meant by a PQ type voltage-gated calcium channel. Quite simply, it's a voltage-gated calcium channel where the alpha-1 subunit is CAV2.1. Okay, similarly, for N-type, it's just CAV2.2. Okay, so if you use CAV2.2 instead, you end up with what's called an N-type voltage-gated calcium channel. So basically, in the axon terminals of neurons, you have voltage-gated calcium channels which will open in response to depolarization, and therefore will open in response to an action potential, because the upstroke of an action potential involves a lot of depolarization. Okay, you have these PQ type and N-type voltage-gated calcium channels in axon terminals, and all that means is that the alpha-1 subunits that you've used to build these things are either CAV2.1, which gives you the PQ type, or CAV2.2, which gives you the N-type voltage-gated calcium channel. Okay, right, so that's all that means, basically. Okay. The important thing to understand about that is that we have these channels which will open in response to depolarization and allow calcium into the cell. Okay, so basically, when an action potential arrives in the axon terminal, it's going to trigger depolarization here. That will open this voltage-gated calcium channel, and I should just put the VGCC here, okay, bringing in calcium into the cytoplasm of the axon terminal here. So we have now turned an electrical signal into a calcium signal inside the cytoplasm of the axon terminal. Okay, what we now want to study is how does calcium trigger the removal of the complexin? Okay, and you might think, well, does the calcium just bind directly to the complexin? No, not directly. It binds to another protein, which then causes the release of the complexin. Okay, so we've got one more protein to discuss. Okay, and the protein that we now need to discuss is another protein, actually, that is present within the synaptic vesicle membrane. Okay, and this is a protein called synaptotagmin. Okay, so synaptotagmin is a protein within the synaptic vesicle membrane, just like synaptobrevin 2. So I might go back to my picture up here of the synaptic vesicle membrane and draw my synaptotagmin in this picture here. Okay, now, synaptotagmin once again is an integral membrane protein, but it's oriented the opposite way. The amino terminus is inside the lumen of the synaptic vesicle, then you have your membrane spanning alpha helix, and the C terminus is going to be extracellularly. But before I show you the C terminus, I need to show you two important domains of synaptotagmin here. Okay, which are C2 domains. Oh, and I should just stress, synaptotagmin is not a snare protein. It doesn't have a snare motif. Okay, that's a confusion that some people have. Okay, it's not a snare motif though. It's not a snare protein though. Okay, its important domains that it has are C2 domains, those domains that are capable of binding calcium, remember. Okay, so you call the two C2 domains of synaptotagmin the C2A domain and the C2B domain. So remember, C2 domains were those ones that we saw in protein kinase C. Okay, they were beta sandwiches, and in the loops between the beta strands in the beta sandwich structure, you could occasionally bind calcium, and those loops were called calcium binding loops. Okay, and that's the basis of how C2 domains can bind calcium. So in synaptotagmin, you have two C2 domains, two beta sandwiches in tandem like this. Okay, the first one is called the C2A domain, and the second one is called the C2B domain. Okay, so in these synaptic vesicles then that are primed like this, ready to fuse if only you could remove the complexin protein, you have these synaptotagmin proteins. Okay, and when a calcium signal occurs in the cytoplasm, And what's going to occur is that calcium ions are going to bind to our C2 domains, and I actually do know the number of calciums that bind to each of the C2 domains of synaptotagmin. Okay, 2 calciums bind to the C2A domain and 3 calciums bind to the C2B domain. Okay, so in our synaptic vesicle, we've got lots of synaptotagmin proteins. When the calcium signal occurs, because an action potential has arrived in the axon terminal, the synaptotagmin proteins are going to now start binding to calcium. Specifically, each one is going to end up getting 5 calcium ions bound to them. Okay, and one of the key things that's then believed to occur when these synaptotagmin proteins get these calciums bound to them is that they bind to syntaxin 1, basically. Okay, they bind to a component of this core snare complex here. Okay, and when they bind to the snare complex, it's believed that they then cause complexin to break off, basically. Okay, releasing the clamp. Then what happens is these snare motifs of this snare complex can finish the job. They can finish zippering right up, and when they finish zippering right up, they bring these membranes so close together that they're then forced into fusing. Okay, so they become the same membrane. So just drawing a little picture of this, it goes from being like this to being like this, basically. And then all of the neurotransmitter that's stored within the synaptic vesicle here will be released into the synaptic cleft. Okay, and it can then go and act on the receptors in the postsynaptic membrane. Okay, so that is an absolutely key physiological example of where calcium signals from extracellular fluid calcium is really, really important for causing changes in the behavior of cells. Okay, this is how all neurons all over the body are releasing neurotransmitter, basically, in response to these calcium signals. Okay, so obviously it's an extremely important example of calcium signaling in physiology. Okay, so that now concludes my discussion of calcium signaling.